Hey guys, welcome back. This time around we're going to take a look at the muscle contraction at the macroscopic level. And that's also known as the gross level. If you recall in previous videos, we've actually taken a look at muscle contraction at the microscopic level. There we said we had a contractile unit known as a sarcomere. Here we're going to take a look at how all those sarcomeres coordinate to result in what we think of as a muscle contraction, such as the biceps or triceps contracting. At the macroscopic level, we see we have a contractile unit known as a motor unit. Now, a motor unit is going to be equal to the motor neuron plus all the individual muscle cells it innervates. Now, let's back up just a moment and try to describe that. The motor neuron is going to be issued from the spinal cord, travel via the ventral root towards the effector organ. In this case, the effector organ is going to be a skeletal muscle. As it gets close to that skeletal muscle, it'll start to branch. At the end of these branches are going to be what's known as axonal termini. Now, these axonal termini are what's going to directly communicate with the muscle cell. Depending upon the number of branches that we find associated with that motor neuron will determine the number of individual muscle cells it controls, because every axonal terminal will have one and only one muscle cell fiber that it innervates. Innervates means creates that neuromuscular junction between the end of the axon and the muscle cell itself. So if I have a muscle neuron that branches and innervates and has, let's say, 100 axonal terminals, that means that it will control 100 muscle cell fibers. So each time an electrical event comes down that axon, we will see that it will actually trigger electrical events on the surface of 100 different muscle cells that will result in a muscle contraction, coordinated muscle contraction, hopefully. The number of axonal terminals also tells us what type of muscle contraction that neuron's controlling. If it's a fine, delicate type of muscle contraction, say your finger's picking up a feather, then we're going to need to be able to control only a few muscle cells. But if you're looking at a leg muscle, that would be different. In a leg muscle, for instance, you may have several thousand axonal terminals for a given neuron. So when that neuron fires, it can control a large number of muscle cells and have more of a gross movement kind of approach associated with it. We can measure the series of events that occur macroscopically during a muscle contraction using a piece of equipment known as a myogram. A myogram will measure the different phases of that contraction. Depicted here would be the results of a single stimulus. That is called a muscle twitch. And when we are looking at the muscle twitch, we are looking at the response of the muscle to a single action potential on its motor neuron. And it has three phases. One of those phases is the latent period. Another one of those phases is the contraction phase. And the third phase is the relaxation phase. Let's first see what's happening during the latent period. During the latent period, it will be the time at which the stimulus has been initiated, but before we have any cross-bridge attachments occurring. Now think of all the things that we discussed so far that occur between the moment that the stimulus begins and we have our first cross-bridge attachment. That would include all the electrical events coming down a neuron, followed by the transfer of that electrical event from the neuron across the synaptic cleft to the postsynaptic terminal, to the localized depolarization that will result in an action potential, which traveled down the T-tubules, triggering open the voltage-regulated calcium gates at the sarcoplasmic reticulum, the efflux of calcium in, binding to the troponin, causing the tropomyosin to roll out of the way, exposing the myosin binding site on the active molecule. All those events occur during the latent period. Notice how brief it is. And then we have the contraction phase. Now, the contraction phase is when all those events that we looked at of the cross-bridge attachment, pivot, detachment, cocking, Now, the relaxation phase is the time in which we take the muscle cell, and in reality the nerve cell as well, back to its original state. Now that means that we've got all that active pumping of sodium and calcium back to their original millimolar concentrations on a given size of the cell. You can also see from this diagram the events of polarization, depolarization, and repolarization played out. The only thing that I would identify differently from this diagram is the realization that what is in green here is repolarization occurring. And finally, notice that depending upon the muscle cell, the duration of the twitch will differ. If the smaller the muscle motor neuron is, the shorter the contraction phase will be. Think of the extraocular muscle, in which we're taking a look at a motor neuron that's controlling very few muscle cells versus those of the soleus, in which we're going to find that one motor neuron controls thousands upon thousands of individual muscle cells. Now, as you are aware, we normally do not display a muscle twitch as a muscle contraction. If truth be told, muscle twitches frequently are annoyances to us. Instead, our muscle contractions are smooth, long, with the strength varying depending upon the demands of the body at that given moment. This means that what we will actually have is something known as a graded response. Now, that graded response can occur one of two ways. It can occur by taking a single muscle and changing the speed of stimulation. Now, here you can see wave summation being played out. Let's look at wave summation. What happens if we have a second electrical event before relaxation completely is satisfied? Can we have that second electrical event result in a second muscle contraction? And the answer is yes. The limiting factor is that electrical event, that repolarization that's occurring during the relaxation phase, has to be able to go past threshold towards repolarization. Once that's happened, then the second electrical event can occur and bring us back across threshold. Now, the neat thing there is notice that second electrical event, while it's the same strength, that second electrical event results in a stronger muscle contraction. Why? The answer is that, remember, for the muscle contraction to occur, we must increase the level of calcium inside the muscle fiber. Now, during a muscle twitch, all the calcium that was released from the sarcoplasmic reticulum is reabsorbed back into the sarcoplasmic reticulum. But during this phase in which we're looking at wave summation, between the first and the second electrical event, not all that calcium was taken back up by the sarcoplasmic reticulum, meaning that when the sarcoplasmic reticulum is triggered by the second electrical event to release calcium, it's got to release less calcium to start the contraction because there's already calcium present in the cell. By adding more calcium inside that cell, it can cause more cross-bridge attachments to occur. So notice it gets stronger, we're building up momentum until we reach that magical point called tetanus. Now, tetanus is not tetanus that you think of as a locked muscle in your jaw, but tetanus is when we see the maximum contraction rate realized for a given muscle. Now, this can continue as long as we can provide ATP to that given muscle. Once we run out of ATP or become deficient in ATP, notice that cliff, it falls off the cliff there. No matter how much electrical event we have, it will not result in another muscle contraction. In fact, at the end of that peak there, we have something that we would think of as a contracture. Contracture we frequently call a cramp. Now, where would this type of muscle contraction that would be beneficial to us? It would be beneficial for those muscles that we are going to exercise in a short period of time, a short window, and then stop and have an opportunity to rest in. That could be those muscles that you're developing associated with activities such as playing basketball or tennis. So you can see tetanus has its pros and cons. Pros are it can bring a motor unit to its maximum strength quickly. But the con is that it's very fatigable. It depends heavily upon the ability to retain oxygen. So it might be that we want to find a different way in which we can sustain an event over a longer period of time, but also a way in which we can control the strength of the response depending upon the need. An example of that would be, let's think about it. Say your index finger and your thumb, and you can use it to pick up, those two fingers to pick up a variety of things. Let's say that you're picking up a feather. Do you want to have the same pressure on that feather as you would if you were trying to pick up a larger rock? I don't think so. I think the larger rock, you'd want a stronger response than if you were picking up the feather. How can we grade those types of response based upon need? Well, the way we can do that is we can activate motor units based upon the need. Every muscle will have a variety of different levels of motor units. There may be 10 motor units in the muscles of your fingers, for instance. Some of those motor units may have a motor neuron that is innervating only 1 or 2 muscle cells. Another cluster of them may have that are innervating hundreds of muscle cells. And then there could be those units in between in which you have motor neurons that have 10 to 20 or 30 muscle cells associated with them. So what I can do is recruit the number of motor units and the type of motor unit based upon the response that I need to give to the given stimulus. Picking up a feather, I may only activate the motor units that have only a few muscle cells. Picking up the rock, I may actually activate the motor units that have only the few. Then recruit those that have an intermediate number of muscle cells that they control. And then if I need to, I can then also ask the larger motor units to kick in. So I can have a graded response. I can grade the number of motor units that I bring online depending upon the demand of the stimulus. Nice thing about that also is that at any given moment, I've always got a few motor units that are at rest while the others are contracting at maximum. I can take those that are fixing to get fatigued offline at the same time that I bring on the others that have been in reserve. And so I can switch them out sort of like we do with our armies, so that we can make sure that we can we can give the ones that have fatigue rest while we're bringing the ones that are rested online. This is something known as recruitment, multi-motor unit summation known as recruitment. And it is, of the two, the most valuable one to be using if we're using those muscle contractions over a long period of time and need to sustain them. Now this finishes our discussion of gross skeletal anatomy. Now it's time for you to back up. Take a look at the muscle anatomy. Take a look at the contraction, excitation, and the excitation-contraction coupling events. And weave the skeletal muscle contractions at the gross level into those bits of information. Hope this video helps. See you in the future. Have a good day.